And here we go with one more video on muscles. Nice way to start, right? Seeing a warning sticker. I will just tell you now, just for the record, that this is a challenging topic, and you will probably have to watch this video a couple of times at least before it's going to sink in. But you can do it, and we'll talk about it some more in class. But I'm just letting you know now that you can't just breeze through this video and think you got it, because this might be some of the most involved anatomy that we're going to do in A&P. So let's just go ahead and get started. It's not going to be that bad, just relax. But all we're going to focus on in this video is how is skeletal muscle organized and how do the parts of the skeletal muscle interact to produce a contraction. So how is muscle organized? I think if I give you an analogy, it makes a lot more sense. Okay, this is a drinking straw. A drinking straw is wrapped in its own individual wrapper, right? Okay, so far so good. I could have a box of individually wrapped drinking straws, okay? And then I could have, let me move, I could have a crate that contains boxes of individually wrapped drinking straws. Okay, so think what's inside of this crate. Boxes. What's inside of the boxes? Wrapped straws. If you think about that analogy, and I'll come back to it on the next slide, this should make a little bit more sense. This whole thing, as we established in the last video, is an organ. It actually contains connective tissue and muscle tissue, and you see some blood vessels here, and you're seeing the bone nearby. So let's walk through what we mean by our analogy. If you look right here, this thing that's pulled out that we can see here, this is called a muscle fiber. This actually is a muscle cell. Wait, I thought you said fiber. Muscle fiber is the term for an individual muscle cell. Okay, each individual muscle cell is wrapped in its own wrapper called an endomysium, kind of like the fiber is the straw, and endomysium, this little white layer around here, represents the endomysium. Awesome. I could have a bunch of these individually wrapped muscle fibers, all wrapped in their endomysium, in a box, in a unit, and the unit is called a fascicle. And the fascicle has a different kind of connective tissue wrapping around it, or at least the term is different. This is called a paramysium, and we said before, whenever you see mys, we're talking about muscle, para, around. So now we have the wrapping around the individual muscle fibers. But I have these, here's a fascicle, here's a fascicle, here's a fascicle, here's one. I can have all of those wrapped in this tissue. This is called the epimysium. So this represents the crate. So let's go back. Here's my drinking straw. The straw is wrapped with an endomysium. My box of wrapped straws is called a fascicle. The box itself would be called the paramysium. And then all of the wrapped fascicles are wrapped in another layer of connective tissue called the endomysium. I'm sorry, not that one. The epimysium is here, and the epimysium represents this connective tissue that holds everything together. And then notice how all this connective tissue eventually will join a tendon. And we know that tendons, again, are connective tissue, of course. And since they're all going to be going this direction, when that muscle contracts and pulls on all that connective tissue, it's going to pull on that tendon, pull on this bone, and the bone is going to move. Okay, so most tendons are like this. They're going to be these straps, if you will, that hold muscle to bone. There are some exceptions, and I want to show you this one exception over here. An aponeurosis is a broad flat tendon, and that's going to be what you see here. You see what's called a palmar aponeurosis. It's just a modified tendon. It's going to be a flat tendon rather than one that looks like this. But if this analogy doesn't make sense, go back to the straw analogy before you keep going. You want to understand this piece before we move on to the next slide. So assuming you do, we'll move on to over here. Now, here's where we have to get into some pretty involved anatomy because ultimately the goal is to understand how a muscle contracts. We're going to focus on skeletal muscle. That's what you see up here. And notice how we have these dark and light bands in it. Once again, the term for that is striated. It's a striated muscle. So this is a skeletal muscle. And you're seeing the individual lines here of dark and light. And that's kind of the stripedness, and we'll see where that comes from in a second. We see nuclei here indicating it's multinucleate. So let's look at the diagram and kind of make all this make sense. Okay, what you're seeing here, this whole thing is a muscle cell. Again, we call muscle cells muscle fibers. So what do cells have? What do you remember that cells have? Cells have parts, like they have a nucleus. In this case, they're going to have more than one. Here's another one. They're going to have mitochondria. And what do mitochondria do? They're important for the production of ATP. Do you think muscles are going to need to have some mitochondria? Sure, they're going to need to have a lot of ATP produced for contractions. Muscle cells, all cells, have membranes. The membrane for muscle cells, that is muscle fibers, is called a sarcolemma. So rather than a plasma membrane, the muscle fiber has a special name for its membrane, a sarcolemma. And rather than cytoplasm being kind of the goo that's all in here, we call it sarcoplasm instead, just because that's how A and P people are. Okay, so mitochondria, ribosomes, all the other cell parts you would think would be in a cell are in the cell. You do see these dark and light bands. You know, here's a dark band, here's a light band, and we'll talk about the origins in a second. But one significant thing that you see in here is these little pieces called myofibrils. Okay, these are going to be special units that make up a muscle fiber. And again, don't call them fibers because it will just confuse you. This whole thing is the muscle fiber. These pieces are basically organelles. They're going to be units within that muscle cell, that is muscle fiber. These are the myofibrils. These are why we have these dark and light bands. So let me go ahead and show you this. Let me move. So if I were to blow up this section here and look very carefully at what I'm seeing from here to here, I'm going to see this is a blown up version of a myofibril. And the striations, striated, are due to the patterns of filaments within the myofibrils, and we'll look at those in just a second. But notice, if I were to look right here, I got orange lines and blue lines, and all of this together is going to look kind of dark because it's very dense. Where it looks kind of light, I'm going to have the appearance of a light band. Where I got all of this stuff again, where I've got orange and blue together, this is going to appear dark, this is going to appear light, this is going to appear dark, this is going to appear light, this is going to appear dark, and that's why muscle fiber is striated. Let me get rid of my drawings here. So when we look at this, we see the dark and light bands. And how do you remember this? The dark, the A band is the dark band, the I band is the light band. Why do you have those? It's because of the arrangement of two particular protein types. You have actin and myosin, and you'll see them better in just a second. But right now, let's focus on this idea that the reason we have this dark and light pattern is because the arrangement of these filaments within a myofibril. Myofibrils are parts of muscle cells. Muscle cells help make up muscle tissue. Muscle tissue helps make up the skeletal muscle. Whew. And we're not done yet. We'll be okay. Okay, so here's a blown up version of what we just saw, same thing. Okay, and I've actually indicated for you a unit called a sarcomere. A sarcomere is a contractile unit within the muscle. And notice it goes from the middle of a light band all the way across to the middle of the next light band. So this is a sarcomere. Here's the next sarcomere. You can't see it all. It goes from here all the way over to there. Here's the one going this way. It goes from here over to there. So a myofibril has all of these sarcomeres. I think of them like train cars. They go back to back to back to back. And those are going to be the things that get shorter during a muscle contraction. You will hear me talk about sarcomeres a lot because that's a really, really important unit. So the sarcomeres have these boundaries. We refer to the boundary between a sarcomere and a sarcomere as the Z disc. Z just means the end of. So here's one sarcomere. So you can say this is the end of this sarcomere, and it's also the end of the next sarcomere right next to it. So we have the Z disc. Again, we have the I band, that is the light region, because we only have one kind of filament in here. Where we have multiple kinds of filaments, this is the A band or the dark band, and then we have again over here the Z disc for that particular for that division between the sarcomeres. You don't have to worry about the H zone unless you want to. It's just the very center of the sarcomere, but we'll make sense of that in just a second. So that's the sarcomere, but let's add a layer to it. If we were to look a little deeper and really kind of look at this more carefully, we're going to see this, where we have these orange pieces. Okay, those are these kinds of filaments. We mentioned two kinds of filaments before. This is the myosin filament. This is the orange one. And that's, again, this corresponds to this. This and this are the same thing. Okay, sorry. The thin filaments, these right here, correspond to these. Okay, so again, make sure you see that connection. There's one other kind of filament we won't focus on too much called titin. Those are these kind of little springy ones. We're not going to focus on those too much. You really need to focus on the thin filaments, actin, and the thick filaments, myosin. So once again, we see the Z disc and the Z disc. And again, study these pictures until these things make sense. You have to see the sarcomere in there. Okay, one more level to this. If I were to look in here, and let's pretend I could be a little person. I'm now going to try to draw a little person. I could be a little person, and if I was going to look right through here, what would I see? I would only see, for the most part, and again, forgetting titin for a second, I'm only going to really see this, right? I'm only going to see these thin filaments. So that's how that view would look to me. If I was kind of be able to look through it, I'd only see that. If I were to look just right here, I'm only going to see the thick ones. Yeah, if I were to look right here, I'm only going to see thick filaments, which is why I get that view. If I were to look right here, I'm going to see, again, you see accessory proteins. They're not really shown on this, but I'm going to see a little bit of a linkage. But if I were to look right here, I'm going to see dark and light, dark and light, dark and light, and I would get that overlapping region. I give you that view just to get you oriented on what it looks like, because what we have right now is we're going to have ultimately we're going to have a movement of these filaments. And if you can see how right now there is no actin right here, so there's actin here but no myosin. Here there's actin and myosin. Here there's only myosin. Then you'll start to conceptualize how a muscle works. Okay, so here's one more even deeper view of this, and you see these pieces. This are the thick filaments. This is the myosin. And notice how they look when you really zoom in. They're like little golf clubs, like somebody took a bunch of golf clubs and twisted them together, and you have these little pieces here. These are called the myosin heads. Okay, you also have the thin filaments. These are the thin filaments that look like twisted up pearls or twisted up beads. Okay, so once again, we have where they're overlapping here. Ultimately, what we're not seeing, of course, is here and there representing the Z discs, the end of the sarcomeres. But here in the center of the sarcomere, you're only going to see, it says the thick filaments like myosin heads. Myosin heads are only present in areas of overlap. Don't worry about that detail now, but a little bit later we'll make sure that makes sense to you. So again, this is a sarcomere, most of a sarcomere. If I was trying to draw more accurately, I would draw this. to show you how the thin filaments are coming in here. And I'll come over here just for completeness. So this whole thing is a sarcomere, but it has actin and myosin, and eventually what's going to happen is going to be a movement of these filaments. We call this idea of how muscles contract the sliding filament theory, because filaments slide. Okay, we're almost there. Let's see, where did it go? There you go. So one final look at this, and then we'll wrap up, is, again, we're looking here at a sarcomere. We are seeing the thin filaments, the actin, the thick filaments, the myosin, and now we're going to pull them apart separately so you can see just a little more detail. The myosin, again, has these heads, and then it has these tails. The actin has a couple other proteins that are associated with it. It has a protein called trypomyosin and troponin. So trypomyosin and troponin are going to be really important. Trypomyosin is probably a better way of saying it. Trypomyosin and troponin are going to be really important for how myosin interacts with actin. And I'm going to kind of spoil it for you now and tell you, ultimately, myosin is going to grab actin in particular locations and pull on it. And that's how we get the contraction we associate with skeletal muscle. So that will be coming up in class. So to start to sum this up, okay, again, this was the muscle. When we look at, again, this is the muscle fibers here. There's two fibers shown. But now when we zoom in on the actual muscle cell itself, the muscle fiber, we see the components of muscle. In particular, we see the myofibril. The myofibril is going to be composed of all of these repeating units called sarcomeres that are made of actin and myosin. Actin and myosin are these proteins that are going to be interacting to produce a muscle contraction. There's a lot to absorb here. Please take your time and go through this. Okay, just one more view of the same thing. This is just showing you kind of a pulled out version. Here is this whole thing is a muscle fiber. Here is a actin and myosin filament within a particular myofibril. You see the sarcomeres represented here as those dark and light bands repeating over and over again. You see the other components of the cell. And there's some things that are mentioned here that we didn't get into. We will in class. You see, for example, and I lost my marker again. We see the transverse tubules, and we see sarcoplasmic reticulum. We'll talk about those in class. But kind of the final questions to absorb here